Okay, good morning, friends. As you know, we deal with your elective, that is 6M elective, that is plant disease management. I hope you have already done with your core. And in the next 6 classes, that is starting from today, every day we have a class from 7 to 8. We have, 6 classes allotted for this elective. And in these 6 classes, each class, for a duration of 60 minutes, that is 1 hour, 7 to 8. And in the next 5 days, that is including today, in 6 days, we'll be covering the entire, plant disease syllabus, plant disease management syllabus that has been prescribed for your 6th sem. As you know, this, what do you mean by the disease? Disease is dis-ease. Ease means comfort. When you are comfortable, that condition we call ease. When you are uncomfortable or discomfort, you face discomfort, or when you face any kind of deviation in terms of physical or physiological, that leads to what we call disease. So any deviation, either in the structure or in the function, or any deviation or discomfort experienced by any organism, maybe a human being or a plant. This deviation we can see, in the form of what we call discomfort. If you observe very carefully, you will be in a position to identify the discomfort. See, for example, if it is human being, just by looking at your friend's face, you can say, see, you're looking very dull. What is the matter? Or by touching his, body, you can say, see, it is quite warm. It is, temperature is more than normal. So, are you well? Are you feeling any discomfort? So is the case with, crop plants. We confine ourselves to crop plants. And what are the major discomforts that crops face during their, reproductive phase, starting from seedling to, you know, maturity stage? At each stage, the plants may face some discomfort. So these discomforts, what we call diseases, and generally in plants, generally we discover these discomforts in the form of what we call, symptoms. They show certain symptoms. So generally these symptoms are visible on various aerial parts. Maybe mostly, whenever we say plants, generally what comes to your mind, that is leaf. The leaves which are green, glittering, turgid, may show certain symptoms, or they may become pale, yellow, droopy, may show, warty structures, or curling, or hypotrophy, etc. Or some cases, wilts. So all these are nothing but symptoms. So by looking at the symptoms, we can recognize that that particular plant or that particular crop is suffering from some ailment or the diseases. Now, these diseases are generally, they are caused by animate and inanimate. Inanimate means not with any biological organism. See, for example, even human beings also suffer from diseases, not only from biological organisms, but also abiotic factors. See, for example, now you know how is the weather outside? It's very hot, very scorchy. If you happen to move in the scorch or very hot sun, you are likely to develop, or you fall, uh, you know, uh, into what some kind of discomfort or some disease, that is heat stroke, you suffer from. So if you go to very cold areas, say for example minus degrees, say if you go to Kashmir or if you go to Canada or American or Europe, where very chilly weather, that is minus 10 degrees, minus 15 degrees. Under such inclement weathers, our body cannot tolerate such too cold conditions. So we get indisposed. So these are environmental conditions. Now, so for example, there are certain conditions, you and me suffer from, say, diabetes. We suffer from what we call hypertension. People suffer from paralysis. Okay. So these are nothing to do with any causative organism. There is no organism responsible for it. Such diseases are inanimate or environmental. Say, for example, heart diseases, if you sit with a heart patient, if you live with a heart patient, you don't get any heart problem. If you sit with a paralytic person, you don't get paralysis. Okay. If you sit with a person suffering from, diabetes mellitus for a number of days, you don't get. It's not communicable. It is not, contagious, because there is no organism, inoculum responsible for that particular ailment. Unlike this, there are so many diseases which are caused by so many organisms, biological organisms. So not only plants, animals, every living being may suffer from one or the other ailment. Now these elements are diseases caused by certain pathogens. Whenever we speak of disease, you come across what we call a pathogen and a host and a parasite. Host gives shelter to the pathogen, and pathogen, it lives on the host and causes a disease. So like this, there is a causative organism. For every disease, there is an organism. And for our convenience, we divide these. Of course, you're quite familiar with this, just brushing or make you recapitulate whatever I'm giving these few minutes as an introduction. So now a number of organisms are there. Say, for example, starting from a virus, a bacterium, a fungus, a mycoplasma, a protozoan, a worm, a roundworm. So these cause a number of diseases in our crop plants. They are known to cause diseases in crop plants. Thereby, plant suffers heavily. Plant suffers heavily means farmer also suffers heavily because of that. So the main intention of introducing this plant disease management in your syllabus is you are supposed to know what are the common diseases that are encountered in our crop plants and how best you can protect your crop plants. What are the various ways of this disease management, protection mechanisms? So you will be knowing in our next 5 classes about what are the common diseases that are encountered in our important crop plants. So in crop plants means we discuss about your cereals, we discuss about millets, we also discuss about what we call pulses, we also discuss about what we call cash crops such as cotton, sugarcane, etc., etc. And also, diseases caused by your viruses, bacteria, mycoplasma, fungi. In addition to that, we also discuss about diseases or discomforts caused by other plants to the important, economically important plants. There are certain plants, which live as parasitic mode on other plants. Angiospermic parasites we have, what we call the parasites are of, root parasites, stem parasites, complete parasite, the partial parasites, etc., etc. So this is about a brief thing. So here, your syllabus, the syllabus, entire syllabus has been divided into 4 blocks, and each block, there are 3 units. And like that, we have in all 12 units. We have, 6 classes. In each class, we will be, covering 2 units. That way, we'll be covering, at the end of the 6th class, we'll be covering all the 12 units or the chapters that are there in our syllabus. So we have, first, major block is what we call major disease of crop plants. Major disease of crop plants. Under this, uh, primarily we discuss about in today's class, fungal diseases. Fungal diseases 1, fungal diseases 2. And in the second class, we'll discuss about what we call bacterial, viral, and mycoplasmal diseases. And also thereafter, we'll also discuss about various pests. See, the diseases are caused or losses are caused not only by fungal, bacteria, and viruses, but even pests, that is insect pests, they too cause a lot of damage to our crop plots. They not only cause damage to the standing crops, but also they cause a lot of damage in your godowns. If you buy, pulses or rice, wheat, etc., on a large quantity, and if you fail to open and observe them, in 3 or 4 months, you will see development of a lot of pests. They cause a lot of damage to our stored products. So we'll discuss about various pests that cause a lot of damage to our storage products and also on the standing crops. Thereafter, the block 3 and 4, we confine or we devote our time towards how to check, how to manage these, diseases and pests. Disease and pest management. So under this, we'll discuss about what are the various concepts of this pest management and what are the important principles that are involved in pest management. And under this, we'll discuss about, various cultural practices, various physical practices, various chemical methods, various biological methods, etc., etc. And in addition to that, we'll also discuss about the recent methods of these disease and pest control methods. This includes what we call an integrated pest management, not one, but all. So the managerial practices you use for one crop may not be fit for the other crop. So we have to look at each crop, requires a definite method of pest management. So all these various physical, mechanical, chemical, biological, all these put together are called what we call integrated pest management. And also we'll discuss about latest methods of this pest management, especially the transgenic technology. Whenever I say transgenic, of course, you are well aware of it. People coming from, villages and having a little knowledge of what we call agriculture, they know various seeds. You might have heard about what we call BT seeds, BT cotton, BT brinjal, etc., BT mustard nowadays. So these BT is nothing but Bacillus thuringiensis, a transgenic plants. So we'll discuss about this integrated pest management. So this is broadly our syllabus. So now today we will rush through, because it needs, at least for a detailed coverage, we need at least 15 classes. But, we have, very little time and we have to rush through. And what my advice to you is, because you have the material in advance with you, and you are supposed to go through the topics which we are going to deal to in next day class, tomorrow or day after class. So what my advice is, try to go through them and come with your doubt. So these online and offline classes are meant for, clearing your doubts, clearing your doubts. Okay, so now we will discuss about fungal diseases. See, fungal diseases, so one thing you have to remember. Of all the pathogens that cause a lot of damage to our crops, the damage caused by fungi outnumbers, outweighs when all other pathogens put together. That is, loss caused by fungal pathogens is far more than the loss caused by your bacterial, viral, and other pathogens. So we have, we will discuss about in today's class about what we call various diseases that are very prominent and that cause a lot of damage to our crop plants. Especially, we'll deal with three important crops. The first one, as you know, is paddy, that is rice. We'll also discuss about the wheat crop and important wheat diseases. We'll also discuss about diseases observed on maize. We'll also discuss about the disease that is noticed on groundnut, even on cotton, even on cucumbers, even on what we call bajra, etc., etc. So we'll begin with rice. Rice is the staple food of South India, or for that matter, more than 50% of the world population depends on rice. Rice is the staple food, even in India also. In South India, our staple food is rice. So rice or the paddy or Isa sativa, what we call, it suffers from various fungal diseases. So the important fungal diseases of rice are, one is what we call blast of rice and brown spot of rice. And the third one is what we call foolish seedling growth of rice or Botany disease of rice. These are the three important diseases which we will concentrate on. The first one is, the blast of rice. Why the name blast? So the infected crop, the infected field, if you look at an advanced stage, the entire crops, appears as if it is burnt. Blast means burnt. So it looks as if the entire field is burnt. Hence the name blast. The blast of rice is caused by, Magna porte oryzae. The earlier name was Pyricularia oryzae. Pyricularia oryzae or Magna porta oryzae causes this blast of rice. Blast of rice. And the other is brown spot of rice. The brown spot, as the name tells you, on the leaves, you will see brown spots. The lens-shaped spots surrounded by, brown halo. And hence it is called brown spot. This brown spot of rice is otherwise known as helminthiosporosis. Helminthiosporosis. See, this disease, this occurred, Helminthosporium oryzae. The causative organism is Helminthosporium oryzae, and its new name is what we call Drexlera oryzae. Drexlera oryzae is the new name, and Helminthosporium oryzae is the old name. And this brown spot disease of rice, it was so prevalent in 1940s. In 1942-43, you might have heard, Great Famine of Bengal. In Bengal, the staple food is rice. As the entire crop area deltoic area got infected by this Helminthosporium varice or Dressler varice, resulting in total crop loss, resulting in 5 lakh deaths. Hence, it is called Great Famine of Bengal. Thereafter, you know, scientists across the world concentrated on and developing how best we can check the spread of this disease, and what are the various procedures that can be adopted to produce resistant varieties against these pathogens. And the third one is, this Botany disease. Botany is otherwise known as, this was first spotted in Japan. Hori, a scientist, uh, identified the pathogen for this Botany disease. The term Botany in Japan means foolish seedling growth. Botany disease, foolish. Here, the seedling, that is rice, generally grown primarily as a dry crop, mostly as a what we call wet crop, submerged. In submerged, generally what we do, we raise seedlings and they are transplanted. Now these seedlings, some seedlings, the Japanese farmer, in his field, he observed certain seedlings showing overgrowth. Some were just 1 feet, the other was 3 feet. And he named this as a foolish seedling, Botany. And this is caused by what we call Fusarium species, Fusarium fusicori, or otherwise what we call Gibberella fusicori. It was identified as Gibberella fusicori, a sexual stage of this Fusarium fusicori. Now, the reason for this excessive growth or overgrowth, you might have seen, some people are very dwarf, just they are 3 feet. And some you might have seen, some grow more than 7 feet. I think some 5 to 10 years back, there was one gentleman from Nizamabad, I think. His name was Gattaya, who was more than 7 feet. This is because of what we call excessive release of pituitary hormone. So here also, the excessive growth of the seedling is because of what we call excessive production of gibberellic acid. Gibberellic acid, it enhances, the expansion of the cells, hence excessive growth. So these are the 3 important diseases of what we call in rice. Now, first up, the Magna Porta varice, what we call, paddy blast or blast of rice. Now, this is caused by Pyricularia varice or Magna Porta varice. Now, this is an ascomystis, and generally, if you look at the infected crops, you will see what we call white patches. White patches you can see, and these white patches are encircled by what we call brown borders. These white patches, in due course, here and there, 3, 4, spindle-shaped white patches you will see, and they in due course they get circulated by brown spots, and these small spots, in due course they coalesce or they mingle, they join with one another, and the entire leaf becomes grayish or whitish. And once the leaf becomes whitish, its photosynthetic ability gets reduced, and the panicle may not be, you may not see the panicle, and even if you see the panicle, you will see the seeds are half filled or unfilled. So a lot of damage will be there. So this it mainly reproduces by what we call, by conidia. And you will see the conidia coming, and the conidia are unbranched conidia, and these conidia bear at its tip what we call the conidiophore bears lot number of conidia, which are obclavate, or what we call, a reverse club shape or top shape. Each conidium, which is 3 to 4 celled, and these conidia are produced in a single spot, in a single spindle shaped spot. You will see production of 20,000 or more than 20,000 of spores. So such large number of spores are produced. They are very light, and if there is a gust of wind, if there is a rain, they get carried away. So they form the inoculum for the development of the disease in the healthy plant. So within a week, you will see appearance of these spots with the entry of the fungus into the host. It produces spots within a week or 10 days. And within next week, it produces a lot number of spores, that is conidia, and they get disseminated easily because of their very light. They'll be easily carried away by the blowing wind. Thus, within no time, your entire field gets infected with this disease, that is blast disease. Now, at the end of the season, it reproduces by producing what we call, ascospores in what we call in sexual reproduction by the production of what we call perithecium. A perithecium, wherein you see what we call an osteole, and inside you will see ascus, and each ascus produces ascospores. This is the perfect stage. But mostly the spreading of the disease occurs only through what we call the conidia. Now, at the end of the season, the conidia remain, or the ascospores, they remain on the debris. See, after harvesting, whatever the stubules that are left over, whatever the fallen leaves that are there, whatever the spots that are there, it forms the inoculum for the next crop. So what we have to do, we have to go for what we call cultural methods such as crop rotation, and also we should keep, the bunds and the channels free of weeds, and also because it is also a seed borne. See, even the seeds may have the conidia. So we should treat, hot water treatment, or using dressing with a sulfur, aggressone, cerazone, etc., or copper sulfate, formaldehyde, etc., can be used to check the seed-borne inoculum. Then we will move to the second disease, that is, I told you, helminthosporiosis, or this second disease, that is brown leaf spot of rice. I told you this great Bengal famine was because of this Helminthosporium oryzae or Drexleria oryzae. And here, the difference between that and this is, here the symptoms are eye-shaped, lens-shaped, lenticular, and generally you will see these dark brown spots developing on the upper side initially, and later on you will see on the lower side, and the all these small spots in due course, they are seen appearing on the entire leaf lamina, not only on the leaf lamina, even the leaf sheath, even what we call on the stem, even on the culm, even on the panicle, even on the seeds, etc. You can see this. But here, the seed, here, this brown spot disease, here, even it infects, even seedlings. So even pre-emergence, the seedling may not emerge. Even there's a death of even the seedling will be there. So if the plant survives, when it reaches what we call ear formation, etc., at that time, you will see in these lenticular spots, you will see production of, conidia. conidiophores. And the conidia are, multi-celled conidia. They are 5 to 10 celled, and generally it is more or less, sickle shape. Sickle shaped structures are there. And here, because the conidia are very light, similar to that, blast of rice, even the conidia, they are produced on large numbers, and they get disseminated by blowing winds. So even here also, the damage is very, very large. Here, we have, certain collateral hosts. See, what do you mean by this? If, say, for example, you grow rice. After harvest, you don't see any rice. But, these pathogen or the conidia are known to survive on the collateral host, certain grasses that grow along with the bunds or in the rice fields, such as your Cynodondactylon or Citeria italica or Echinoclea colonum. These are some of the collateral hosts on which the pathogen survives. Okay, uh, so now what we have to do? So to check this, what we have to do, eradication of the collateral hosts, and the sanitation. That is, you should keep your field very clean, and even the stagnation of water should not be there. And even while applying your nitrogen fertilizers, don't apply nitrogen fertilizers at once. Split doses. In 3 splits, you can give the nitrogenous fertilizers. So, and then what the best way is, crop rotation. And the another thing is, go for what we call disease resistant varieties. There are certain disease resistant varieties which are, for example, IR24, Padma. These are found to be quite resistant against what we call brown spot of rice. Then the third one is Botany disease. I told you, Fusarium fusicori, or the other is what we call Gibberella fusicori. This is the sexual stage. And I told you, this was first noticed in Japan, and the infected seedling showed overgrowth. Hence, it is called a fully seedling growth. And now, the excessive growth, the, the seedling dies. Excess of anything is not good. The seedling shows overgrowth and dies. Even if the seedling survives, the plant may show overgrowth, and the leaf, the leaf will be very narrow, and the culm will be elongated, and panicle will be very small, and seeds will be very, very less. So yield gets affected. Lot of loss for the farmers. So here the Gibberella fusicori or Fusarium fusicori, it reproduces by what we call conidia. Here also conidia. In 3 cases, the common mode of reproduction is through conidia. Here, it produces 2 kinds of conidia. Small conidia are known as microconidia, and the bigger ones are known as macroconidia. These micro and macroconidia are produced on large numbers, and at the end of the season, it reproduces by what we call, thick-walled spores called chlamydospores. So through these chlamydospores, it survives, it overwinters and over summering, and it may remain. These chlamydospores may remain in the soil, or they may be seen attached to your seeds. So hence, what we have to do, we have to go for a deep clubbing in summer. If the inoculum, if it gets exposed to sun, they may die, and treat the seeds before sowing. Treat them with Axon and Cerason, etc. Thereby, you can There was small disturbance. Uh, so now we move to the next crop, that is wheat. Now, wheat is also another staple food across the world. Even in India also, if you go to North India, the staple food is uh, wheat. Wheat also suffers from, in our syllabus we have two important diseases that are commonly found on wheat. One is black rust or stem rust of wheat. The other is smut of wheat. Smut of wheat. These are the two important diseases that are there in our syllabus. First one is stem rust or black rust. So here the term rust, what do you mean by rust? Here the symptoms are rusty and the spores are rust-like. Hence the name rust. See, the two important basidiomycetes symptoms. One is rust and smut. Rust are rusty and smuts are black soot like structures. So on wheat, we have both, that is rust and smut. The rust, as you know, you read about this, stem rust or rust, what we call, it is caused by, Puccinia graminis triticale. Uh, and in all the wheat growing areas, you come across the uh, prevalence of this disease. This is caused by, as I told you, Puccinia graminis. And this Puccinia, it is what we call, it is heterogeneous macrocyclic fungus. Heterogeneous means it needs two hosts. One is wheat, the other is barbaris. Wheat is the primary host and barbaris is the secondary host. And it is macrocyclic. What do you mean by macrocyclic? It produces, in its life cycle, it produces 5 types of spores. One is pycnospores, ichthyospores, iridospores, teletospores, basidiospores. As it produces 5 types of spores, it is called macrocyclic. And as it needs 2 hosts for its completion, hence it is called heterogeneous. Heterogeneous. Now, of all the stages, the dangerous one is what we call iridosaurus stage. Iridosaurus stage, and the rusty streaks, rusty spots are seen on the leaves, on the leaf lamina, not only on the lamina, even on the sheath, even on the stem. You can see even on the seeds also, you will see even on the culm, even on the ear, you can see a panicle. On the panicle also you will see these rusty spots. The rusty spots are because of the production of iridospores. Now iridospore is what we call, it's a round binucleated acuminate spore. I know it causes a lot of damage. So the entire crop, leaf becomes rusty in nature. When the leaf becomes rusty in nature, the rust like, its photosynthetic ability gets reduced. So the plant may not flower. Even it flowers, the panicles may have few number of seeds. Even if the seeds are there, if the plant gets affected during milky stage, the seeds will be, or the ears will be half filled. So at the end of the season, iridospore slowly changes to teletospore. The teletospores are, bi-celled, binucleated cells. And because at the end of the season, the iridosaurus changes to what we call teletosaurus. The teletospores are blackish, hence the name, stem rust or black stem. The leaves show rusty pustules, where stem at the end of the season shows black spots, hence the name black stem. Black stem disease. So here, generally in all the wheat growing areas, you will come across this macrocyclic disease caused by this Puccinia. So now the other is what we call smut disease. The smut disease of wheat is caused by what we call Ustilago triticale. Ustilago sagitta. The Ustilago causes, Ustilago is a smut. Puccinia causes rust disease, whereas the Ustilago causes what we call a smut disease. The infected ear, if you look at the infected ear, you will see black soot. The what we call the ovary gets transformed into black sooty sorus. So the seed gets, or the ovary gets filled with this black sooty spores. They are teletospores or chlamydospores. So when the crop gets infected with this, the entire ear transforms into a black soot, and drastic reduction in uh, yield. The yield could get, it could be reduced to by what we call 80 to 90%. The spores are black and sooty, and because they have the ability to survive even in the soil, or they may get entangled to the wheat seed. So when you sow these infected seeds, from the seedling stage itself, the spore germinates and the mycelium enters into the seedling, and as the seedling grows, the mycelium, it spreads throughout the body. When it reaches a flowering stage, then they enter into the ear. Thereby, the ovary gets filled with the mycelium, and the mycelium are the cells. They transform into these, what we call chlamydospores or teletospores, and the all the seeds are replaced by this black soot. Uh, these teletospores are chlamydospores, and farmer suffers heavily because of the infestation of this. So here, what we have to do is how to protect. The protection could be three, four ways. One is cultural methods, wherein we see various agricultural practices by deep ploughing or by ploughing during the summer, etc., and hot water treatment. Of course, if the seeds are infected with these spores, you can get rid of them by treating with hot water. And then what we call chemical method is dressing with, before sowing, treating the seed with agrason, serason, that is wet, dry seed dressing, or wet seeding with, sodium hypochlorite or, copper sulfate, etc., can be used. And the best way is using disease-resistant varieties such as what we call Kalyan 227, PV18, and WG307. These are the, smut-resistant wheat varieties that can be used to check the disease. Okay, this is an account of important diseases of what we call fungal diseases that are very common in rice and wheat. Then we move to the fungal diseases that are encountered in what we call in millets, and the fungal diseases that are found in cucurbits, and also in grapes, and also on groundnut, and also wilt of cotton. So we will deal with um, um, first of all, smut of sorghum. So sorghum, as you know, sorghum vulgare, jowar, as we call, sorghum suffers from smut disease. Smut. So here the smut is black sooty spores. So here the pathogen is what we call the Sphacilotheca. Sphacilotheca, there are three important species causing three important symptoms, diseases. For example, covered smut, Sphacilotheca sorghum; loose smut, Sphacilotheca cruenta; head smut, Sphacilotheca aurelia. So these three species, important species, cause three important diseases in sorghum. So covered smut, to repeat, grain smut or cover smut is caused by Sphacilotheca sorghum. Loose smut, so the ear looks, loose. In covered smut, the seed place is occupied by the smut, and it shows, a white papery covering. And unless and until you disturb that during thrashing, when harvest is done on the thrashing threshold area, when you beat, to separate the seeds, then the sorus gets disturbed. Then spores are released in covered smut. There is a loose smut, the spores are easily disseminated by the air. Whereas in covered smut, it has to be broken. The sorus has to be broken. So now head smut is caused by Raeliana. Here, the entire head, that is entire ear head, it shows leafy structures. Leafy structures, that is your, stamens or the stigma or the glooms. They get transformed into leafy structures. The entire head or the cob becomes, shows the black sooty and leafy structures. Hence, it is called head smut. So, covered smut, loose smut, and head smut. All these three are caused by Sphacilotheca species. So, here, this can be checked, this can be checked by treating the seeds because it is seed-borne. Seed-borne, so Capton and Thyram should be there, and disease-free seeds should be there. Crop rotation has to be done, and thus the heads, the diseased heads, they have to be separated. Either they have to be burnt away or they are buried deep in the soil. And the best way is go for crop rotation. Don't sow repeatedly the jowar in the same field. By doing this, you can check your, prevalence of this disease or recurrence of this disease. Then we go for what we call Bajra. In Bajra, it suffers from what we call green ear disease or downy mildew of Bajra. Penicitum typhoideum is the scientific name of Bajra, and it suffers from Sclerospora graminicola. Sclerospora graminicola is the pathogen that causes green ear disease. Why the name green ear? If you look at the green ear of the Bajra, the infected, the upper part will be normal, and from the lower part, the infected, the fungus, the fungus after entering, it in response to the entry of the fungus, various parts of the flower, that is your globes, your stamens, your stigmas, they become leaf-like. So the entire ear, instead of bearing the pearl-like seeds, pearl millet, what we call bajra, instead of bearing pearl-like seeds, the head shows leafy structures, small leafy structures. So entire, either entire cob or the ear or half of it gets transformed into leaf-like structures. Hence the name green ear disease of Bajra. So this is caused by Sphacilotheca. This is caused by Sclerospora graminicola. Sclerospora graminicola. It is a soil-borne disease. So the best way is growing for crop rotation and treating your seed treatment at a dry dressing or wet dressing, etc. And the crop rotation is there, and going for what we call disease-resistant varieties and burning, separating infected parts, either burning them or burying them. By this, on the best way is using resistant varieties such as HB1, HB15, PHB10. These are some of the disease-resistant, green ear disease-resistant varieties. So, if you notice these symptoms in the early stage, the best way is you can go for what we call spraying of mancozeb, etc., or Capton, or Ridomil, etc. By spraying that, we can check the spread of the disease. Then we move to what we call powdery mildews of cucurbits. This is a common disease, powdery mildews of cucurbits, not only cucurbits, not only in cucurbitaceae, but also even in pulses also. For example, in green gram and even in black gram also, even in vegetables also, you can see watermelon and you know pumpkin and in bottle gourd, etc. If you observe very closely, the leaf exhibits certain powdery patches. So these powdery patches, blotches or patches are seen. And if you look at them, you will see that as if somebody has poured some ash or powder over the leaf. But actually, that powder is nothing but innumerable number of, conidia produced by the pathogen. The pathogen here the pathogen is Podosphaera xanthi or Erysife chicorariaceum. So Podosphaera xanthi and Erysife chicorariaceum these are the two important genera that cause a powdery disease on cucurbits. And these uh, conidia, the conidia are produced in large numbers. Conidia are produced in innumerable numbers. And each conidium is very small. And as they are produced in large numbers, they form as if somebody has poured powder on the surface of the leaf. Hence the name powdery mildew. And when the conditions are favorable, if generally the fungal disease spread, uh, when uh, when the humidity is more, when the humidity is around 80-90%, and when warm nights are there, these are very congenial for the robust asexual production of these conidia. And because the conidia are very small and very light, they get disseminated by the blowing wind. And within no time, the entire field gets affected by the fungus. And we have another disease, what we call in grapes. This is what we call downy mildew of grapes. In grapes, here also the symptoms are seen on the leaf, not only on the leaves, even on the, grape bunch, even on the young fruits also you can see, even on the petioles also you can see downy growth. The downy growth is nothing but, cottony growth. You might have seen cotton, cotton threads. So these, you on the lower side of the leaf, if you look at carefully, you will see as if some cotton is hanging. That cotton is nothing but, that cottony outgrowth is nothing but the conidiophores and the conidia. The conidiophores and the conidia are produced on large numbers and they show what we call downy growth. Hence it is called downy mildew of grapes. And it is caused by Plasmopara viticola. Vitis vinifera is the scientific name. So it is what we call Plasmopara viticola. Now this is unlike that, this is a mastigomycotinum. It produces, the conidia germinate into what we call when the dew or when raindrops are there, the conidia germinate into, the conidium becomes a, a gametangium and it produces gametes. A sporangium, sorry, spores. These spores are, biflagellate spores are there and they swim for some time and later on they lose their flagella and they germinate by a germ tube. So here the Plasmopara viticola causes a lot of damage. The downy area, downy growth, when initially downy growth and later on, the leaf becomes yellowish and leaf becomes, pale and, you know, it shed. So when there is a lot of shedding of leaves, naturally its photosynthetic capacity gets reduced and the grape branch, the grape that is fruits, those berries are of small size. Even on the in the young one also, you can see these downy growths. Even on the fruits also, even on the leaf also, even on the petiole also, even on the young tender stem parts also get affected by this downy growth. So here, the best way is, say, to check this, the famous uh, uh, chemical, that is what we call uh, the Borax mixture. France, once it suffered from this downy growth of Plasma parabeticola, all the grape vines, were got devastated, and the Borax mixture was developed. The borax mixture is nothing but copper sulfate and calcium hydroxide. If you spray that, you can check the proliferation of this disease. Then we have what we call corn smut. And this is smut, as you know, caused by Ustilago maydis. And here, the corn smut, the infected ear, the seeds, these seeds are seeds replaced by what we call boil-like structures or tumor-like structures. You will see tumorous growths. And if you break that tumor, you will see black soot. So the entire ear or the cob, all the seeds get transformed into black sooty structures. And these blocky structures, are nothing but these galls are tumors. Hence, it is called a smut disease, a tumorous disease. And hence, if you disturb that tumor, you will see black spores. Black spores are, they are called chlamydospores or teleospores. They are produced in large numbers. So the cob becomes deformed. It gets deformed and produces many warty or boil-like structures because of what we call hypertrophy, excessive expansion of the cells. The excessive